Good evening, everyone. We'll wait for 3-4 minutes more, then I'll start the session. I hope I am audible. You can reply yes in the chat box.
Ma'am, your voice is very low.
Okay. Can you hear me clearly now? Hello. Okay. So I'll just wait for a minute more, then I'll start. So someone is asking about the recording of the second session, sorry, the first session. So I'll just post the YouTube link on the chat box, the link of the channel. So I posted the link of the channel on uh, on the chat box. If it ever happens that you miss any session, you can go to that channel, and every week after this session, like the problem solving sessions which are being taken by me, those videos will be uploaded there. There are two more TAs in the course, but they'll be uploading their respective videos on their own channel. So I'll start the class now. Good evening, everyone. I welcome you all to the week 2 problem solving session of the NPTEL course Cell Culture Technologies. And I'll request you all to please mute your mic if you have any doubts or any questions in between. You can always put them in the chat box, and at the end of this 1 hour session, if time permits, I'll take up those questions. Even if you have any doubts when I'm explaining any answer or something, you can just put it up in the chat box. So I'll be taking mock questions today. That is, I won't be taking the current active assignment of week 2 of the course. So I'll be taking questions so that you get a fair idea of how to solve those questions. So the first question is, one of the major roles of carbon dioxide in cell culture is to, first option is imbalance pH, second is balance pH, third is balance surface tension, and fourth is imbalance surface tension. So basically, carbon dioxide in our cell culture facility acts uh, to maintain the pH of our cell culture systems. For example, carbon dioxide also helps in buffering the pH of our blood. So here, the correct answer will be, the major role of carbon dioxide in cell culture is to balance the pH. So, very briefly, I'll explain to you all what is the bicarbonate buffering system in cell culture. So, what happens is you have your incubator where we keep our cells in petri dishes or flasks. So, here usually the common practice is to supply carbon dioxide from outside and maintain its percentage somewhere between 5 to 10% of CO2. So this is the most common percentage of CO2 which is maintained in any cell culture laboratory. Now here what happens is CO2 is present in the environment in the outside system, and from there when it combines with H2O or water, and this is the reason why it's equally important to maintain humidity in our cell culture incubator. So once CO2 combines with H2O or water, it forms carbonic acid. Now, this carbonic acid can then further dissociate into H plus ions and bicarbonate ion. And this is the buffering system which takes place. That is, there is a constant interchange between formation of carbonic acid and then this dissociation into H plus ion and bicarbonate ion. Now, if this H plus ion increases, then what will happen? Your pH will decrease. pH will decrease and it will lead to formation of an acidic environment. However, if your H plus ions decrease, then your pH will increase and it will lead to formation of an alkaline or basic environment. Now for cell culture, it's important to maintain the physiological pH, and that physiological pH is somewhere around 7.4. So if it happens, so here this is a equilibrium system where whatever carbonic acid is being formed is getting dissociated into H plus and bicarbonate ion, and also this again is combining and forming this carbonic acid. However, if it so happens that the percentage of your CO2 increases or decreases, then this buffering system will be disturbed, and because of that perturbation in the buffering system, what will happen is your H plus ion will either increase or decrease, leading to basic or acidic environment. But we do not want that kind of environment for our cells. We want our cells to grow in a physiological pH where they'll be the most healthy, and for that we have to maintain this pH 7.4. So in cell culture, the media which is commonly used uh, that is DMEM. So this is the most commonly used medium in cell culture. And when I say medium, I mean this is the medium which provides nutrients, amino acid, growth factors to your cells to help them to grow. So in DMEM, mostly we add an indicator that is phenol rate, and this phenol rate is a pH indicator. And this DMEM is a CO2 dependent medium. So in this CO2 dependent medium, which is DMEM, this pH indicator phenol rate is added. Now here you can see what are the different colors of phenol rate at different pH. So for example, at pH 6.5, it is yellow in color. At pH 7, it is orange in color. At pH 7.4, it is orangish reddish in color. And at pH 7.8, it is pinkish in color. And as you go on increasing your pH, that is basic pH, it will start turning purple. Now, why is the need for having a pH indicator? So what happens is, if like just by looking at the color of your plate, if you see that the color is something like this, that it is orangish, then it is reddish, then you can assume that your cells are healthy and the environment is fine because it is somewhere at the physiological pH where your cells can grow. However, if it so happens that you go and look at your plate and you see that the color has turned yellow or the color has turned purple, then it will be a major alarm, because then if it has turned yellow, that is if it has turned acidic, then that is an indication for contamination in your cell culture. Contamination, or maybe your culture is very old, and uh, if your medium has turned basic, like if has turned purple, then maybe your CO2 supply is very low, and that is the reason like it has turned basic. If your CO2 supply is very low, then your buffering system is not working, it is collapsing, and that is why you need to check your CO2 supply. Uh, are you not able to hear my voice properly? Someone just messaged that my voice is lagging. If you are able to hear my voice properly, can you just reply a yes on the chat box?
Yes, ma'am.
Okay. Okay, then I'm assuming it's fine. Okay. Okay then. So this is the bicarbonate buffering system or the CO2 buffering system, which is used in cell culture, and that is to maintain or balance pH. So this is how the buffering system takes place. Your CO2 combines with H2O or water, and then after that this carbonic acid is formed, as I explained earlier. Now in the media that I was talking about, DMEM, this sodium bicarbonate is added from outside. So the reason for adding it is, when you add sodium bicarbonate and it combines with water, it dissociates into this sodium carbonate and your bicarbonate ions, and This bicarbonate ion will help in maintaining uh, the H plus ion concentration and the pH of your system, because if only CO2 is being supplied, then it will take some time for this H2, this carbonic acid, to dissociate into bicarbonate ions and H plus, and that is the reason this sodium bicarbonate is also supplied. So moving on to the next question. Entry and exit of a lab should be clearly defined and not obstructed to ensure: first, safe exit in case of emergency evacuation; second, safe entry of external air in the lab; third, aesthetic appeal of the lab; fourth, so that nobody can enter the lab. Now, it's very important to define the entry and exit of your lab or your cell culture lab, and you must have listened to this week's lecture of Menak sir, and there, like, if you had listened to it carefully, you will guess that it is important to define the entry and exit of the lab, so that if there is any emergency evacuation, then we can have a safe exit. For example, in case of a fire or in case of a gas leak, if the entry and exit of the lab is clearly defined, like without panicking, we can have an emergency evacuation. So the correct answer is A. Next question. Aseptic techniques are employed in cell culture lab: first, to contaminate cultures; second, to prevent contamination of cultures; third, to remove dust; fourth, for aesthetic appeal of the lab. Now, what does aseptic technique mean? So, aseptic technique is something which maintains sterility. So, it's very important to take into consideration the fact that while you're handling cell culture or while you're working in a cell culture lab, you maintain proper sterile environment, or and you follow aseptic techniques, and that is to prevent contamination of our cultures. So here the correct option is B. So I'll discuss in brief why is it important to maintain aseptic techniques and what do those aseptic techniques involve. So here, as you can see, this is what a common laminar cell culture hood looks like. So here, as you can see, in this box, we can see our clean space. Clean space is where all our sterilized equipment is present. Then next comes your working space. So the working space is the area where your hands are going to be, and you are going to be opening these bottles, handling this pipette, and stuff like that. So an important thing is to that while you are working in this area, your hands should be sterile. That is, you should have wiped your hand with 70% ethanol. You should be wearing gloves, and so on. So these are some very basic aseptic techniques which should be followed not just in cell culture lab but while handling anything in the lab. Then comes your third area where you can put your waste, whatever culture waste, as you can see it's labeled here, liquid waste, or whatever dry waste is going to be. So as you can see here, there's a clear demarcation of the space between your clean area and your dirty area. So in a laminar cell culture hood, this should be clearly defined and your clean area should be separate from your dirty area, and in between can be a working space, because you will be requiring some things from this clean space where your sterilized equipment is present, and you may be discarding some of your used waste in this dirty area. However, it's not a good practice to keep all your sterilized equipment along with your waste, liquid waste or trash or whatever it is, because what will happen is this will also contaminate your sterilized or clean equipment. So this clear demarcation between your clean area and dirty area is important, and this is a very important aseptic technique in cell culture. So next, what are the like areas from where your contamination can arise? So the very first area is your lab equipment. If your lab equipment is not properly sterilized, then you're going to put all this equipment inside your hood, your pipettes, your media, your centrifuge, or anything which you are going to put inside your hood. If it is not properly clean, it's not aseptic, it's not sterilized, then what will happen, this will be a major source of contamination, because this is what which is going to be inside the hood, and from here if any bacterial or fungus spore is present on any of these surfaces, then it is going to contaminate your culture too, because you are going to handle that culture inside this hood. The next source of contamination can be your cells itself. Suppose you revived an old stock, and from there your cells are already contaminated with bacteria or fungi. Then what will happen? If one plate inside the, so this is a cell culture incubator. So even if it happens that just one plate inside the incubator uh, has gotten a contamination because of your already contaminated stock, then it may happen that it's going to contaminate all the plates and all the culture which is present inside the incubator. So your cells or your cell stock or your media for that matter can be the second source of contamination. The third source of contamination can be your cell culture regions. Cell culture regions can include your serum, medium, amino acid solutions, or any solution. So all these things are going to go inside your laminar flow hood, and from there they can further spread that contamination. So it's extremely important to keep all these things sterile and to check for contamination from time to time. Now, the fourth major source of contamination can be the people who are working inside the lab. Suppose you are putting your hands inside the cell culture compartment inside the hood, but your hands are not sterile. You are wearing gloves which are old, which have already touched some surface and have gotten contamination from there. So, like it's important to sterilize our own hands. It's important to wear gloves. It's important to wear lab coat, so that like the staff does not spread contamination. So these are the common sources from where contamination can arise. Now, what are the different kind of contaminants? So next we'll discuss what are the different kind of microscopic contaminants which will contaminate your cell culture. So the first can be virus, and since viruses are very small, it's going to be very difficult to visualize them even through light microscope. So the common light microscope that we use in cell culture we won't be able to visualize viruses through that light microscope. And the next, so even if you get a viral contamination in your cell culture, you will see that your cells are dying, but you won't be able to exactly troubleshoot just by looking under the microscope that what is the source of contamination, because for visualizing viruses you need electron microscopes. Then the next source of contamination can be mycoplasma. Mycoplasma contamination is very dangerous because it quickly spreads to other cultures as well. But with mycoplasma, you can there are some characteristics of mycoplasma contamination through which you can predict that this indeed is mycoplasma contamination. But again, for detecting it and from removing it, it's a whole lot of trouble. And the most common contaminants are bacteria and yeast. So these are the two most common contaminants in the cell culture lab. Mycoplasma contamination is not that common when we compare it to bacterial contamination. So bacteria you can easily see under light microscope, and also just by looking at turbidity of your cell culture plates, you can predict that this is a bacterial contamination, and it's easy to remove bacterial contamination. And bacterial contamination doesn't spread that fast unless and until like you deliberately put the contaminated culture in some other cell culture. And so this is a scale of sizes where you can appreciate the relative sizes of different kind of microscopic contaminants. And this is our normal eukaryotic cell, which can be somewhere around 10 to 100 micron. So here, this is just an example. So look at this picture, this first picture, where you can see that the person who is handling the cell plate, look how that person is handling the plate. Like he or she is touching the plate, not like he has not put his finger inside the plate. Yeah, he's touching the plate through the edges, and he's using a sterile micropipette. And all this is happening inside the hood. However, if you look at this second picture, then you can see that the hood is here, but that person has taken out the tray, and then like taken out this holder, and then whatever he's handling it outside the hood. So this is something which should not be done. Like this person is spraying ethanol on the holder in order to sterilize it, but it is not advisable. Even if you have to keep your ethanol bottle, you should keep it inside the hood, like inside the hood, and there you should sterilize it. Whatever the common equipments which are being used in the cell culture laboratory, they should not be used outside the hood. Like you should avoid bringing them outside the hood. So this is a common example. Moving on to the next question. So the next question is, which of these is used in cell culture lab to store cell line vials for long term? First is liquid oxygen, second is liquid carbon dioxide, third is liquid nitrogen, and fourth is liquid hydrogen. So I think I discussed this in the last class. So you can answer in the chat box. Yeah, so here the correct option is C. We use liquid nitrogen for storing cells for long term. So as I discussed earlier, this is something which is called cryopreservation. And last time I discussed in detail that this cryoprotective agent DMS or glycerol is used for cryopreservation, and that is because it prevents the formation of ice crystals inside the cell. And I also discussed that when you are storing, so this is what a common vial of like common cell vial looks like, and this is the vapor phase of the liquid nitrogen which is used for storing the cell. So last time I mentioned that in liquid nitrogen, broadly there are two phases. One is the vapor phase, and second is the liquid phase. So when we store our cells, we do not store our cells directly in the liquid phase. So this liquid phase is the phase where the temperature is minus 196 degrees Celsius, which is the temperature of liquid nitrogen. However, the temperature is slightly less in the vapor phase. It is somewhere between minus 150 to minus 176 or 178 degrees Celsius. Now, this is a schematic diagram of a liquid nitrogen tank, and this is how, so these are the racks which are present. So in your cryo boxes, these are called cryo boxes. So in these cryo boxes, you keep your cryo vials, which look something like this, and then it is strongly recommended to store your cryo boxes and your cryo vials, which have your cells, in the vapor phase only and not in the liquid phase. And that is why the liquid nitrogen tanks which store liquid nitrogen, there the tanks are filled only till one fourth, only somewhere like one fourth of the capacity, so that when you are storing your cells, they stay in the vapor phase and not in the liquid phase. And here, so this is again a schematic where you can see the broad temperature distribution. So in the lid of the liquid nitrogen tank, so this is the lid, this is a liquid nitrogen tank. So in the lid of the liquid nitrogen tank, the temperature is somewhere around minus 150 degrees Celsius. However, in between the temperature varies, like between 150 degrees Celsius to minus 178 degrees Celsius. And then at the lower bottom where your actual liquid nitrogen is present, there the actual temperature is minus 196 degrees Celsius. But as you can see here also, your cells should be present in these upper compartment in the vapor phase, and it should not be immersed directly into the liquid phase. In some industries, what happens is, instead of keeping the cells like instead of maintaining a boundary like this in the same tank, what happens in industry is they have a liquid nitrogen tank which is fully filled with liquid nitrogen, and they have some other tank connected to it where only the vapor of the liquid nitrogen is going, so that even by any mistake the cells do not get submerged in the liquid nitrogen itself. Because what happens is, if the cells are directly submerged in the liquid nitrogen in that medium, then your cells will not be good for long. In just 1 month, they will be dead. So, for long term storage, for cryopreservation, it's always prudent to store your cells in the vapor phase. Now, moving on to the next question. HEPA stands for, so first is high efficiency particulate air filter, second is high efficiency primed animal, third is health and emergency patient accessibility, fourth is high efficiency primary flow. So I can see some answers. The correct answer is A. HEPA stands for high efficiency particulate air filter. So I'll explain in brief the working principle of HEPA. So HEPA filters work through a principle of physical filtration, physical or mechanical, you can say. So what do I mean by mechanical or physical principle of filtration? So here what happens in your HEPA filter, there is a mesh of fibers as you can see here, and these fibers are a mesh of different kinds, like they have different diameters. All these fibers, they are not of the same size or the same diameter. For example, you can see there is one kind of fiber which is like somewhere around less than 100 nanometer, then more than 1 micron, and so on. So what will happen is, since there are different diameters or different sizes of fiber and there's a whole interconnected mesh, then what will happen, like different kind of fibers will filter out different kinds of particle. For example, the first kind of fibers will filter out particles which are less than 100 nanometer, and that is something called interception. So they'll intercept, like they'll stop the entry of those kind of particles. And then second kind of like your second kind of fibers, what will they do, they will like whose diameter is more than the first kind of fibers, what they will do, through impact they will stop like particles which are of larger sizes, for example 1 micron. So here you can see this fiber is stopping this small particle which is less than 100 nanometer, and then this fiber, this is stopping this big particle which is more than 1 micron. And what will happen, only air will be allowed to flow through this mesh of fibers, and these particles will not be allowed. And then the third kind of like mechanism is diffusion, where just because there is an entire mesh of fibers, it will be difficult for small particles to diffuse through this mesh easily, and then like go through. So what will happen is, like using this mesh of randomly arranged fibers, there will be like different kind of particles which will be filtered out. So this is, and this is a diagram of the HEPA filter. So there is a continuous sheet of this filter medium. This filter medium is nothing but this mesh of fibers, and then there is an aluminium separator between all the different kind of medium. So what will happen is, suppose this is your HEPA, and then this is your outside air, and this here, this air which is coming out is inside air. So what happens is, like outside air has a lot of contaminants which you can see here in this yellow color, but what will happen is your HEPA will filter it, and your inside air, the inside air which will be coming, it will be free of all these contaminants. So your HEPA filters can provide you air which is somewhere around 99.95% pure. And HEPA is used not in just cell culture systems but also in normal home air purifiers. So in cell culture system, like I'll show you where HEPA filter is uh, specifically installed. So this is your cell culture hood or your laminar airflow hood, and this is where you will be working. So here what will happen, because you will be opening this flap to put your hands to work, what will happen is there will be inflow of air, and from here these are your exhaust filters, and here your HEPA filter is installed. So what will happen? Like whatever air is coming from outside, this HEPA filter first of all will not allow this outside air to come inside. And how does it do that? So whatever air will be coming out from this exhaust filter, what it will do, it will be continuously filtering this air, and there will be a continuous downflow of air. Because there will be a continuous downflow of air, this air from here will not be allowed to go inside the hood, and because of this, like there will be a continuous downflow of pure sterile air. So this scale here shows what all different kind of contaminants or what all different kind of particles a HEPA can filter. So as you can see, viruses, bacteria, and your other animal cells, everything it can filter, and that is how it is able to provide a sterile environment. So moving on to the next question. Which of the following is not safe to be operated? First is stainless steel. Second is high density polyethylene. Third is surgical equipment. Fourth is glass. So I can see in the chat box people are answering. The second option is correct. High density polyethylene is not safe to be operated. So I'll discuss in brief what is an autoclave and what is the working principle of an autoclave. So an autoclave is a device which is used in order to sterilize laboratory equipment. So a very important concept to know is that autoclave works on the principle of steam sterilization. So it uses steam in order to sterilize whatever equipment you have, and the it works at a specific temperature and pressure, and it goes up to temperature as high as 121 degrees Celsius, and this is how it sterilizes the equipment, whatever space inside the autoclave. So this is a basic diagram of the autoclave. As you can see, there is water at the base, and this water, like when you will give it heat, it will get converted into steam at somewhere around 121 degrees Celsius, and this is how it will the property of the steam is it has a high latent heat, so it will your steam will have higher latent heat as compared to your hot air, and your steam will be able to percolate inside your equipment, and that is how steam provides a better sterilization than air at the same temperature, that is 121 degrees Celsius. So here, if you look at this diagram, your water is present, your heating element is inserted in the like, your heating element is where your water is. So steam will rise from below, it will rise up, and here you will have a grid basket where your equipment or your media will be present, whatever you need to automate. So this is the area where your equipment and whatever you need to autoclave will be present, and once the steam rises, it can it will percolate through your media, through your equipment. And one important thing is, so before you start your autoclave, your air will automatically be present inside. Now what will happen, if you generate steam and then just you wait for steam sterilization, it will be sterilized to some extent, but your air will still be present. So before you start your sterilization, it's important to remove air.
Of air is important because air hinders any kind of sterilization. So for that, what happens is once you have started your autoclave and it starts heating, the first step is to remove air, and that is done through the air removal valve, and air will be removed through the steam. So after 10 minutes, once your steam has formed, you can open the air removal valve, and along with the steam all the air will be removed. Once your air has removed, you can close the air removal valve, and then again your steam will be formed. And after that, like after 15 to 20 minutes, like once your temperature has reached 15 psi, that is the temperature, that is the pressure at which your 121 degrees Celsius temperature will be achieved. So once your appropriate temperature and pressure has reached, after that we should wait around 15 to 20 minutes, because that is the approximate time which has been standardized by scientists, and this is the time during which your equipment and your media will be sterilized or autoclaved. So this is the basic working of an autoclave. Now there are different types of autoclaves which are used. So basically our autoclave works on the principle of a pressure cooker which we use for cooking. So this is a normal pressure cooker type autoclave which is used in small laboratories where you do not have to autoclave a lot of equipment, and only a small autoclave would suffice. So there we use this pressure cooker type autoclave. Then there's this vertical autoclave. So this vertical autoclave is used in big laboratories where they have to autoclave a lot of media and a lot of stuff. Then there is this common laboratory autoclave, which again it's not a vertical autoclave. Then there's this autoclave which is usually used in industries, and then there's this hospital autoclave which is used in hospitals. Now the next question this uh, is the property of cells which are required to relate the cell cultured cells to functional cells in the tissue from which they were derived. So if you wish to relate your cultured cells, like the cells which you are growing in vitro, so I discussed these two terms earlier, in vitro and in vivo. So your cell culture is a in vitro technique. In vivo is when you are dealing like with the organism itself. So in vivo will be within the living organism. So since we are taking the cells out from the organism and then culturing them outside, that is cell culture, that is an in vitro technique. So but if you have to relate your cultured cells to to the tissue from which they were originally derived, so what will be the property? So that will be the property of differentiation. So this is something which was discussed in the week 1 lecture, I think. So that will be the property of differentiation. Differentiation, if your cultured cells are able to differentiate and retain the same property as when they were inside the body of that organism, then those are your true cells which you are culturing. So the next question, using a pH meter, you find the pH of an unknown solution to be 8. How would you describe this solution? So any so again there are four options: strongly basic, weakly basic, weakly acidic or strongly acidic. So here I can see in the chat box people are saying the correct option is B. So it is correct. If we have a pH 8, then it is a weakly basic solution. So this is the like normal pH scale. For your reference, I have included some biological samples and you can see what is their usual pH. So if we have a pH 8, which is somewhere around the pH of our blood, then it is weakly basic, and a strongly basic. Uh, so this is the pH 7, and that is the neutral pH, which is the pH of the water. And as you go on towards a higher pH, so your pH 13, pH 14 will be strongly basic pH. However, your pH 0, pH 1, pH 2, these will be strongly acidic. So the next question is, which light is used to sterilize the air in the laminar flow hood between uses? IR, NIR, UV or visible light? So here the correct option is UV light is used for sterilization of laminar flow hood or the entire cell culture room for that matter. Now I will briefly discuss how this UV light disinfection works. So what happens is an important thing to know is that in UV light also we have three different kind of UV light, A, B and C, depending on their wavelengths. So for disinfection we use UVC. So what happens is this UVC light is the light which is lethal to microorganisms. So from the sun, these three different kind of UV rays are emitted, and this UVC light is blocked by the atmosphere. It is blocked by the ozone layer, and this is the light which is coming from the sun and which is the most damaging. But thankfully, it does not reach us. A prolonged exposure to UVB light can cause skin to darken and it can cause skin cancer, and a prolonged exposure to UVA light can cause cataract and retinal diseases. So this is the range of like the wavelengths, and as you can see, UVC light has the shortest wavelength. So if it has the shortest wavelength, that would mean that it has the highest frequency among UVA, B and C. And it is filtered out by our atmosphere, but for sterilization, for killing bacteria and for killing microbes, we need UVC. So what happens is there are different ways in which UV light causes sterilization. For bacteria and for viruses, mostly what it does, it leads to the formation of ROS or reactive oxidation species. So here what will happen is, because of formation of ROS, there will be oxidative damage to the cell wall, and once your cell wall is damaged, like your bacterial cell will burst. Same with viruses, there is oxidative damage to viral proteins or to viral capsid, and because of that the capsid or the envelope degrades, and this further will lead to nucleic acid degradation. Another very important point, like another very important mechanism through which UV causes direct damage to the DNA is through the formation of pyrimidine dimers, or these thymine dimers. So moving on to the last question, since like our 1 hour is over and Google Meet will like soon close this session. So the last question is, a major goal in embryonic stem cell biology is to develop protocols that enable lineage specific control of First is division, second is differentiation, third is survival, fourth is all of the above. So, if you want to uh, develop protocols which will allow you to do lineage specific control, then you want to have all, like, you want to have all these kind of properties. You also want your cell to show division, you also want it to show differentiation, and most definitely you want it to survive. So here the answer will be all of the above. So this is all for the questions today. Since we have a 60-minute limit, like in 3-4 minutes the session will discontinue, but I'm opening the chat box open right now. So if you have any question, you can put it in the chat box and I'll reply it. So we still have 5 minutes. So you can put your question in the chat box. For people who joined late, I'm again pasting the link to my YouTube channel. There you will find, yeah, there you can find all these recorded sessions. Do we use BSL in place of laminar airflow? Uh, do you mean like the name? So BSL is biosafety level, and most of the people also call it BSL hood or BSL 2 hood. So most of the cell culture labs have a BSL 2 hood. So there are different levels of biosafety, and BSL is basically your biosafety laminar or biosafety hood. Yeah, so Uma Maheshwari, you are asking about today's session, right? So by tomorrow morning, I'll upload this session on on YouTube as well. So you can find this video also on the channel link that I have put in the chat box. Okay, then thank you so much for joining the session. Seems like you do not have any questions. Again, if you have any questions, you can put in the comments section as well. So Dhanush, I am not able to understand your question. Uh, the extra medium remove the waste beaker. Beaker contains water. So are you asking that in the picture that I showed, why is there extra water? So that was not the extra water, that was the used discarded medium. So in cell culture, we have to continuously change media of the cells so that like so that their pH is maintained, and so that whatever nutrients that are being utilized by cells can be replenished. So in the picture, the beaker that you saw, that beaker was not water, that was liquid medium, discarded medium. And even if someone is using water, like beaker having water in cell culture, it may be specific to them, like specific to their requirement. Okay, then thank you for joining the session. The next session will be on next Friday, same timing. You can join using the same link, and you will also get a reminder through NPTEL. So I'll end this class now. Thank you.